From Microbe TV, this is TWiN, This Week in Neuroscience, episode number 17, recorded on April 20, 2021. I'm Vincent Racaniello, and you are listening to the podcast about the nervous system. Joining me today from California, Ori Lieberman.
Hey guys, how's it going?
It's going well. I'm going to say you're in California because that's where you are. You're no longer here at New York at Columbia, right? Are you finished with Columbia?
My graduation is in May.
Yeah.
So I still have another month of Columbia.
Okay.
It's weird after 8 years. So
you gonna miss New York?
No.
Yeah, nice.
No hesitation.
Not a true New Yorker.
I went hiking. Yeah. No, definitely not.
Also joining us from Salt Lake City, Jason Shepard.
Morning, everyone.
And we have now two new hosts. Well, one of them was on last episode, but I wasn't there. But he's right here in New York City at NYU School of Medicine. Tim Cheung, welcome back.
Thank you. Hello. Nice to be here.
And today we have a new host from Nashville, Vanderbilt University, Vivianne Morrison. Welcome.
Thanks. Good to be here.
How's the weather in Nashville? Is it nice?
I mean, it's gorgeous. It's so beautiful. It's just, I don't even know what temperature it is outside, but it's sunny, blue sky.
We don't want to hear about it.
I know. Let me just rub it in.
Rich Condit from Austin, it's always 80, 90, 100 degrees, no matter what time of year it seems. So, all right. And we have today a guest to talk about his work. He is from Yale University, not too far upstate here. Michael Nitabach, welcome to TWiN.
Thank you. Thanks for having me.
Pleasure. And your weather's probably like ours up there, right? Like partly sunny, partly cloudy in the 60s.
Yeah, I'm actually sitting in my house in New York City right now. So
you are? You live in New York, or you just have a house
I
there?
do. No, I live in New York.
And you commute to Yale?
Yeah.
You take the train?
Yeah.
My chairman used to be at Yale, Shankar Ghosh. I don't know if you know the name.
I know the person, yeah.
Yeah.
I don't know him personally.
So I have to tell you something. Years ago, there was a virologist at Yale by the name of Jordi Casals, a very well-known virologist. And he lived here in the neighborhood of Columbia in Washington Heights, and he used to commute also. And the reason that what was cool is he was working with Lassa virus in the 70s when it was first identified, and he was mouth pipetting it at Yale, and he got sick. So they put him in the hospital here at Columbia, and it so happened that they had treated a nurse from Nigeria at Columbia previously, and they had saved some of her serum, and they gave it to him, and he lived. But I always used to think, this guy commutes to Yale from Manhattan, and now I see another person commuting to Yale.
It's not common.
Not that common. A lot of, interestingly, a lot of people live in Princeton. We have a few faculty who are here and they live in Princeton, which is, hour and a half on the train. Some people actually commute to Philadelphia. Anyway, Michael, we're going to talk about your paper today. But before we do that, let's hear a little bit of your history. You've been at Yale a while, right? Where did you start out? Are you from the U.S.?
Yeah, I was born in Philly and grew up in New Jersey, went to undergrad at UPenn, PhD at Columbia, not at the medical school, in the biology department.
With who?
In Eduardo Macagno's lab.
Yeah, sure.
So this was quite some time ago.
Yeah, I know.
And then got my JD at NYU, and then did postdoc training at NYU, and then started on faculty at Yale about 18 years ago. And I've been at Yale since then.
So you're a lawyer?
I am.
And
retired.
Did you ever practice?
Yeah. No, I did practice, yeah. patent litigation, trademark litigation at a firm in New York City for 6 years, 6 years.
Oh, wow.
Wouldn't happen to be Cooper Dunham, would it?
No, it was Milbank Tweed.
Yeah, I know Milbank, sure. All right, we're going to talk about your paper published in Science, C. elegans, discriminates colors to guide foraging. I'm going to hand it over to Jason, right? Is that, or who wants to?
Yeah,
Ori?
and Ori, but I would love to know the backstory about this, Mike, because so traditionally Mike's lab works on Drosophila, and I'm sort of interested to know, what started off this project and how it sort of evolved. Because I think your Dipon, who was the first author, ended up finishing it at MIT, I think it was, or yeah.
Yeah. So, I mean, there's a saga of the science and there's a saga of trying to get your shit published at intersect in this story. But we had been working on C. elegans for a while before this project started, and Dipon had already basically done. And I just should say at the outset, the entire creative, intellectual, and experimental driving force behind this work was 100% Dipon. When he was in my lab, the parts of it that went on in my lab, I just stayed out of his way. And he's been a postdoc in Bob Horvitz's lab at MIT for the last few years, where he basically did the work that turned out to be figure 4 of the paper. And Bob, it was the same thing. Bob stayed out of Dipon's way. And that's the smartest thing that we could have ever done as far as mentoring Dipon as a student and as a postdoc. So we had been working on C. elegans for a while, and Dipon had already done an entire PhD thesis worth of work that he published as a first author in Neuron, and on a completely different type of behavioral paradigm and circuit cracking, whatever. And he just, is a curious person and was looking into the literature on, worms live in rotting garbage where they feed on bacteria that are decomposing the food waste. And so in that environment, worms need to discriminate between healthy, good bacteria to eat and harmful, including pathogenic and non-nutritious bacteria. And so Dipon was just interested in this and was looking into the literature on the food choice behaviors of the worm. And the laboratory model for this behavior has been giving worms the choice between eating E. coli, which is perfectly fine, healthy food for them, versus Pseudomonas aeruginosa, which is an opportunistic human pathogen, but totally ubiquitous and relatively harmless bacterium for people with intact functional immune system. And so there's a whole literature, it's not that huge, but there's probably a couple dozen papers looking at how C. elegans interacts with Pseudomonas versus how it interacts with E. coli, and how it determines that Pseudomonas is harmful, pathogenic, and avoids Pseudomonas in favor of eating E. coli. And the basic gist that's been in the literature for a while is that it's a chemosensory detection of certain toxins that are secreted by the Pseudomonas. So Dipon's looking at this literature, and he had been interested in light-dependent processes in the worm for other reasons, and just noticed that the Pseudomonas, that one of these toxic chemical compounds that are detected with chemosense as a chemosensory cue, happens to be, in addition to a chemically reactive molecule, is also a blue pigment. This is the chemical called piocyanin. And there were at the time maybe 2 or 3 papers, and also everyone knew this when they started shining bright blue light onto worms to do GFP imaging and GCamp imaging, that worms avoid very rapidly and robustly avoid with a locomotor response, extremely bright blue light. So it just occurred to him, well, wow, maybe worms are using the blue color of this pigment as a way of identifying Pseudomonas and avoiding it. And so that was where he started with that pure speculation, and then embarked on the experimental, testing of that idea. And the basic temporal progression of how he figured this out is actually, which is not that common, but in this case, the temporal narrative in reality is the scientific narrative as laid out in the paper in terms of the progression of the data from figure 1 through figure 4, and even the panels within the figures is basically the order in which he actually did all those experiments, which of course is not the norm, right? The norm
that's rare.
In order to test the hypothesis of blah, blah. And it's like, no, that has nothing to do with why when something was done or when it was done. But in this case, it was all a truthful narrative as laid out in the paper.
Yeah, it's often I tell students that if you read a paper and they go, oh, well, how did they come up with that idea? I'm like, well, they probably had 5 ideas and that's the one that worked.
Yeah, I think that's one of the biggest obstacles in training for young scientists is that they expect that they're going to discover things in that order. And when it doesn't happen that way, it's really discouraging. And so I think going forward, people should be told, no, you're going to discover things you didn't even know you were looking for in orders that you never thought were possible.
Yeah, I mean, a lot of the time, hypotheses, I think, are convenient fictions, if you will, that enable an organization of ideas in a way that is then comprehensible by other people who weren't involved in the actual process of exploring those ideas. And, everybody understands, or as you say, in other words, I think students and postdocs need to explicitly figure out, and perhaps, as mentors we can be, and I try to be quite transparent about the fact that these are convenient narrative fictions that we construct to make it easier and more compelling for people to understand what we're talking about, but that it doesn't indicate that was the actual, historical narrative of how ideas get developed.
Yeah, and the danger is that then they try and prove their hypotheses, and they really get stuck on their hypotheses, and that's when it starts to get a bit dangerous, because then they blinker themselves, which is not good. And I think, and then they hear the study sections, they hear about everyone saying you have to do hypothesis-driven science, and then we're neglecting exploratory science, which is equally important. But anyway, I think Ori has a question.
No, I was just going to also say that, and then in my most recent paper, we had 2 figures, which ended up being the last 2 figures for years. And then to save them and actually get them into literature, we did the rest of the experiments. But I was going to ask if we could take a step back. This is the first time we've talked about C. elegans on the podcast. So maybe, Mike, you could kind of talk about C. elegans as a model system. What's the history of its use? And what have we learned in terms of neurobiology and host pathogen interactions from it?
So C. elegans has been in use as a model system since the early 1960s. So Sidney Brenner in the UK, he was one of the sort of founders of molecular biology, of course, and understanding the genetic code and stuff like that, how mRNA is an intermediary between the genes and the proteins. And so he basically was looking for an organism that could sort of serve as like the E. coli, but in the context of developmental biology and neurobiology, not in the context of molecular biology. And so they wanted something that would reproduce very quickly and exponentially, like E. coli, which C. elegans do. So they're self-fertilizing hermaphrodites. And so sexual reproduction and sexual behavior is not a rate-limiting step for generating progeny. And the generation time is a matter of days from the fertilized egg to the adult laying eggs. Very small number of cells in the entire animal. So the other thing that made C. elegans transformative for developmental biology, and as it turns out also neurobiology, is that it has a completely invariant cellular lineage from the fertilized egg to the complete mature, fully post-mitotic in the somatic tissues adult. So there's like 900 or some cells in the entire adult hermaphrodite worm, and every single cell can be identified and has a name. They're not like names like Fred and whatever, but they're, code names actually mostly referring to their lineages, because it's a fully deterministic lineage of each adult cell from the fertilized egg. So that made C. elegans extraordinarily powerful for asking questions about developmental biology, such as cell specification, tissue patterning, etc. And then from the neurobiological perspective, also, of course, those general developmental questions arise in neurobiology. But from the standpoint of behavioral neuroscience and systems neuroscience, trying to understand the circuit basis and the computational infrastructure for sensory motor transformation and control of behavior, having completely defined, identified neurons that can be studied in multiple individuals of the species, but each time with each individual, that you're looking at and or manipulating the exact same neuron with the same synaptic connections, same neurochemical identity, et cetera, is extremely powerful for systems neuroscience, of course.
And then, so what are kind of like the seminal discoveries, if there are any in particular, that have come from C. elegans that we wouldn't have been able to get without C. elegans?
Yeah. So, well, wouldn't be able to get is not a place I'm going to go. I don't think that's how science works. I mean, we don't need to get philosophical and epistemological here, but I'm not going to say it like that because I don't buy that. But the key important discoveries that have arisen out of C. elegans from a neurobiology perspective, most importantly, and this was a Nobel Prize that was awarded to DuPont's and several others, but including DuPont's postdoc mentor, Bob Horvitz, was the role of cell death in the establishment of the adult structure of the nervous system, and the fact that in this invariant lineage, there are pre-programmed deterministic cell deaths that occur in the lineage that basically terminate certain branches of, if you will, of the, lineage of the nervous system. And so using the hugely powerful genetics of the worm. So because you can grow worms in mass quantities, and they're very easy to image the cellular structures, it was possible to do forward genetic screens looking for mutations that cause aberrations in those patterns of both lineages per se, but also cell death events. And so that really cracked open, from a genetic standpoint, the pathways, cellular and molecular and genetic pathways that determine development of the nervous system. And since we now understand from genome sequencing that all metazoan multicellular animal genomes all derived from a single common ancestor. Of course, these pathways, like the apoptotic cell death pathways, cell lineage determination pathways, are highly conserved between worms and other animals, including obviously human beings.
And then, of course, there's the connectomics.
Exactly.
Because worms only have a couple of hundred neurons and they're very stereotyped, the wiring diagram was, the first wiring diagram of a nervous system was worked out in C. elegans, and that was sort of Brenner's pet project for a long time, and then taken on by others later. And that's been the precursor for the current connectomic projects that are even now ongoing in flies and mice.
Exactly. And I mean, one of the things, this doesn't really relate to this paper that we're talking about today, but one of the things that I realized pretty quickly once we started working with C. elegans is that connectomes, so, maybe the worm is a little bit idiosyncratic in the nature of its connectome, which is, other than the motor neurons and the sensory neurons, there are actually synapses almost between everything and everything. So you can clearly identify, if you look at the network, you can clearly identify motor neurons and sensory neurons, because the sensory and idiosyncratically, sensory neurons receive synaptic inputs and motor neurons form synapses on cells other than muscle fibers. But other than the sensory neurons and the motor neurons, the interneuron networks are highly interconnected. And so when you make these hypothesis wiring diagrams saying, well, we're going to test the hypothesis that we derived from the connectome, that this cell is connected to this other cell, to the other cell, blah, 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 that's really fictional, because you could, just from the connectome, you could have posed probably dozens of equally likely hypotheses up front, and no one ever tests all of them, right? It's not an unbiased, hypothesis generating tool. And I think that that's how the connectomes seems that the fly connectome is turning out to be that way as well, that it's important and useful to know what's connected to what, but it certainly is not telling you how anything works or even giving particularly specific insights into the flow of information, honestly.
Yeah, I'm with you there. I'm skeptical about what principles you can get out of the wiring diagram. And
you need the info, though, right?
It gets exponentially harder to figure it out.
Get to a point, yeah. Once you get to a point where you have a cell that is an important, way station for a certain type of information, you need to know what its inputs and outputs are. Anyway, I don't know that tangent.
Actually, but I feel like that.
Sorry, Ori, in case we're moving away from the connectome, I have a quick question concerning the connectome. So one of the reasons people hypothesize that things are highly interconnected with each other is so that they can be redundant, so that you have a way of, if something fails, you have another pathway to take. So if C. elegans are very interconnected with the interneurons as well, does that mean they are very robust to destroying certain cells, or are they still sensitive?
Yeah, I mean, I think that's a lazy explanation for this high interconnectedness, particularly for an organism like C. elegans. I mean, these are self-fertilizing hermaphrodites. They can basically survive and reproduce, under the very favorable conditions as a laboratory. They can survive and reproduce without any nervous system function at all. Like none. So as long as their pharynx can pump, muscular pharynx can pump and suck in bacteria and grind up bacteria with its spontaneous muscular activity that doesn't require neuronal input, then the animal reproduces. It can't lay eggs. The eggs grow inside the parent hermaphrodite and hatch inside and burst out. So, for an organism that has that sort of lifestyle, it's not that clear to me how intuitively obvious it is that redundancy in a nervous system is particularly powerfully selected for. So I think what's selected for is actually not redundancy, because it doesn't matter if 99.9% or 99.99% of all worms die without reproducing. What matters is that the few that do reproduce. So I think it's about flexibility, actually, and not about redundancy, and that it's about having built into the nervous system this ability to respond in ways that aren't stereotyped and baked in by the conditions in which everything has been selected for, so that when those conditions suddenly change, you don't kill off the entire population. You only kill off 99.9 whatever percent of the population, and a few animals, live to fight another day. But it's all speculation, right?
You find out about this redundancy, well, not redundancy, this ability of the worms to not need a nervous system yesterday on a thesis committee from Eric Jorgensen's group here. They were basically destroying the nervous system and still being able to image these synaptic proteins in these nerves as they were dying, and the worm was just fine. I was just so jealous.
So maybe you could give us some background of what's known about how an organism that doesn't have eyes and a retina and photoreceptors can see light. Because in that sense, this is kind of like the main crux of the paper.
So we don't know. So there's two different light-dependent behaviors that we understand to exist in the worm. One is this avoidance, rapid escape responses to very bright, short wavelength light. And then there is what TWiN discovered, which is this spectrally dependent color discrimination behavior for light of much more modest intensities. So the work that exists, both prior work on these escape responses and now TWiN's work on the ability to discriminate color, we understand the behaviors to some extent and the psychophysical properties, meaning the physical parameters of the light inputs that drive these behaviors. And we know some genes that when knocked out or have polymorphisms across these wild strains, in the case of the color discrimination behavior, lead to either elimination or alteration of the light-dependent behaviors. What we don't understand is what proteins in the animal, if they even are proteins, what molecules in the animal actually absorb photons to underlie these light-dependent behaviors and then transduce that photon absorption into a biological signal that can be, amplified, channeled, and used as inputs to neural computations that lead to changes in behavior. Whereas in the case of opsin-based, eye-based visual processes, we understand all that very well. We know that it's rhodopsin proteins that absorb photons directly via the retinal chromophore, the conjugated chromophore, and that absorb, the visual cascade is very, very well understood as to how that absorption of a photon by retinal changes the conformation of the rhodopsin, G proteins, and so on and so forth. In the worm, which has no opsins in its genome, although, it has no opsons in its genome identifiable by sequence homology. That isn't to say that there aren't, for example, 7 transmembrane domain proteins in the worm genome of unknown function that may be light absorbing proteins either directly or via some sort of conjugated chromophore.
I had a sort of naturalistic question. I always assumed that worms, or certainly C. elegans, were living in really dark situations. So in this case, you're saying that they can come up and they don't need to live in, dark. They can actually take advantage of naturalistic light.
Yeah. So where they actually, this is the lore, right? Oh, C. elegans, they're soil dwelling nematodes, which basically what you find in the soil, this is work done by an investigator named Anne-Marie Felix, who was basically started looking maybe 15 years ago, 20 years ago, started looking for wild populations of C. elegans. And since it was like, oh, it's a soil dwelling nematode, she was just looking around in dirt and basically finding almost no worms. What she found in dirt were some what are called dower larvae, which is a state of kind of suspended animation that worms enter when times get tough, when energy resources are depleted or other sources of environmental stress get very high. And there's a larval arrest. I forget which larval stage, but there's a larval arrest. Development stops and they become this very, very resistant suspended animation form of a worm that can survive for years without any, additional water, food. They go into a state of quiescence and can live for years. So that's what she found in dirt and soil. And there are nematodes that live in soil and that actually, you see the adults and they're in the soil doing their life business, where she found tons of adult reproducing, active, feeding, locomoting C. elegans wasn't in the dirt, but in rotting vegetable matter on the surface of the earth. Rotten fruits in orchards, human compost heaps and garbage piles, trash, human trash. I mean, C. elegans has spread around the entire world, almost certainly in association with human activities, and is basically a human commensal species of Caenorhabdites nematodes. The C. elegans species is a human commensal. And so they live in trash on the surface of the earth, not down in the soil. They reproduce, they feed on the surface of rotting food, and then they explode their populations until all the food gets eaten out. If there's like an apple in an orchard that a worm encounters, there could be an explosion of the population of worms in that apple until it gets completely depleted. And then they all just die, and a few worms go into that suspended dower state and just sit there in the soil under where that apple was for months or years until, another apple falls down right on top or whatever.
Yeah, it's interesting how these sort of myths of the natural environment get propagated, because I assumed that was the case, that these are soil dirt living worms. But it's I was also actually talking to Eric yesterday about this too, and he was like, no, that's not true. And so, but it sort of rams home this idea that if you don't understand the natural environment the animal is actually living in, you wouldn't know to even ask the question.
Exactly.
Are these worms sensitive to light, or can they use light? So why would you even look at it?
So I'm actually interested. we're talking about blue light in this paper, but like what you were saying about how the C. elegans is living in an environment where there's lots of decaying plant matter, like vegetables and fruit. So those are like really colorful, right? Yellows, greens, reds, whatever. So is there reason to suspect that these worms have an entire array of like receptors, like the ones we're going to be talking about in this paper, that are going to, I don't know also the diversity of pigments that are created by the other microorganisms in these heaps, but I imagine that it's actually quite, there are a lot of different colors.
Yeah. So the answer to everything you've said is that all of what you said seems to be the case. And in fact, Dupont embarked by looking to see if worms could discriminate the presence and absence of blue pigments. And through the, sort of gradual elaboration of his psychophysical experiments, realized that what the worms are doing is actually assessing the balance between short wavelength blue photons and longer wavelength yellow orange that we call amber photons. And then again with psychophysical experiments, so not surprisingly, he figured out that worms basically respond with greater avoidance of bacteria in the present when the balance of blue and amber shifts towards the blue direction. That is, when the light gets bluer, the worms have a sort of heightened reactiveness to the bacterial lawn, as if they're saying, oh yeah, maybe this is bad bacteria, which is what you would expect given that Pseudomonas secrete a blue pigment in bait, which by definition is increasing the blue to amber ratio of photons that the worm experiences. But then his purely psychophysical experiments revealed that if the balance shifted in the opposite direction, that is an increase in amber photons versus blue as compared to white light, that also heightens the worm's sort of like, oh, this may not be a good source of food. And we haven't tested this hypothesis, but it immediately led to the speculation that this would be a way for the worm to also avoid bacteria more readily that are secreting long wavelength pigments, red, orange. And there are common, again, also opportunistic human pathogens, such as a genus of bacteria called Serratia that secrete red and orange pigments, and that also are in rotting garbage and trash like Pseudomonas. So whether worms actually use that color spectral dependency of their laboratory behavior to avoid also not just blue secreting bacteria, but orange and red secreting bacteria is something to test, but is suggested by the experiment. So everything you said is exactly on point.
Can I quickly raise a point regarding what you just said, which is when I was reading Figure 1, when you and the first author discovered that the worms are more sensitive to a toxin when there's blue light shining on them. I was thinking back when I was at school, in primary school, we used to play with these things called Newton's colored discs, where you would have all 7 colors of the rainbow on a disc and you spin it and it becomes a white disc. I don't know whether you guys played with it when you were a kid. But what that suggests is that inside white light actually is contained blue light. And when I was reading Figure 1, I was thinking, well, this is rather strange, because for the worm to be able to tell the difference between white light and blue light, something else has to happen, because blue light itself is within white light. So if the worm is just sensitive to one wavelength alone, as far as the worm's concerned, it's just black and white. It's just grayscale.
Yeah.
So So like I was thinking that it must mean that the worm is actually sensitive to two different wavelengths and do a comparison between the two. And then in the second figure, you guys found that it is actually also sensitive to amber light. And that made a lot of sense to me. So that's just I just want to point that out.
Yeah, no, I mean, and we were also, at the beginning sort of, kind of waving our hands up, because it's exactly what you said. In other words, if you only have one spectral channel, then you can't discriminate color, because you can't discriminate between changing the intensity of a, broad spectrum white light versus being in the presence of blue light versus some other color of light. So you need at least two spectrally distinct input channels to do any form of color discrimination at all. We do it with three channels, right? Red, green, and blue. So C. elegans seems to be doing it at least, and who knows, there could be a third channel that we just haven't detected yet, but at a minimum, they're comparing blue and amber. And by comparing the relative intensities in two different places, two different wavelengths of light, you can then distinguish between an actual change in spectral content versus just a change in the intensity of light, as you said.
Maybe we could get into the actual experiments, Michael. Sort of tell us, how do you do this? What's the experimental design?
Yeah, so the key experimental design, and this isn't something that we came up with, but the key behavioral paradigm is that you grow bacteria in a little lawn on a petri dish, and then you just put worms onto that lawn, excuse me, and you see if they stay in the lawn or if they leave the lawn. And that's, and you can just count over time the fraction of worms that stay in the lawn versus leave the lawn, presumably because they don't like the lawn and they're looking for something better. And then within that paradigm, DuPont basically figured out a series of genetic manipulations that can be done both on the bacteria in the case of the Pseudomonas and on the worm in terms of the knocking out genes in the worm and seeing how that affects. And then also manipulating the spectral environment, manipulating the spectrum and intensity of light that's shining down on the lawn and the presence and absence of colored pigments put into the lawn, as well as chemical toxins like paraquat. And so mixing and matching and changing all those various combinations of what kind of bacteria is the lawn? Is it Pseudomonas? Is it mutant Pseudomonas? Is it E. coli? Is there chemical and pigments doped in, supplemented into the lawn, and changing the wavelengths and spectral properties of the light, changing all those parameters in different combinations and permutations, basically let him understand ultimately how it is that the spectral content of incident light onto the worm influences its foraging. And all of that is figures 1, 2, and 3 of the paper, basically. And that was all done in my lab at Yale. And then figure 4 was done at Bob Horvitz's lab at MIT, which was using this lawn avoidance paradigm in a highly reduced scenario. So this is now not with Pseudomonas secreting its native toxic blue pigment, but healthy E. coli with a colorless chemical aversive cue at a low level that the worms wouldn't avoid the lawn except when the spectral properties of light are in specific ranges of blue to amber ratio, and then screened in that exact specific environmental context screened a panel of, I want to say 70 or 80 different wild strains of C. elegans that have all had their genome sequenced, and found, it didn't have to be this way, but found that there was a very wide range of degree of avoidance of the lawn in this exact specific environmental context and spectral context. Tremendous variation in the extent to which these different strains of C. elegans, wild isolates, would or wouldn't avoid the lawn. And then because we had because others, the field has developed full genome sequences of all those strains, you can do essentially what is exactly the same as its quantitative trait locus QTL analysis, exactly the same statistical genetics as what occurs, for example, in human genome-wide association studies, which exploit inter-individual genetic polymorphisms and correlate that with phenotypic variation to identify likely genes in which these polymorphisms contribute to the phenotypic variation. DuPont did that with C. elegans for these, for the spectrally dependent behaviors. And what he found were very powerful genetic determinants of what was many things were fascinating about this, but one of the things that was fascinating was an independently determined, powerful genetic determination of the sensitivity to the more amber ratio as opposed to the more blue, the bluer ratio of colored light. So there seems to be genetically independent control of the sort of gain on avoiding bluish versus avoiding amberish. And then he was able to use the statistical genetics techniques to basically home in on specific, very small loci within the worm's genome that determined that most strongly associated statistically with blue sensitivity versus amber sensitivity, and then use forward genetics, generate CRISPR generated null mutants in 5 or 6 different genes nearest to these loci of highest statistical association, and found the ones that, when mutated with CRISPR, actually completely obliterated the sensitivity to the blue versus the amber and vice versa.
Sorry. Any of these genes that DuPont discovered, are they intrinsically photoreactive and changes?
Yeah. So, what the cellular and molecular mechanisms are for absorbing these photons and transducing them into these behavioral decision outputs, again, we have no idea. What we do know about these genes is that they don't encode proteins that are, known or thought to be photoreceptors themselves, but actually are in stress response pathways. They're highly conserved genes present in what looks to be all eukaryotic cells, and seem to play roles in cellular stress response, including, interestingly, cellular stress responses to UV light. So there is this connection to light and a connection to stress response. And so that led to, our speculation that what may be going on in the worm. So, worm, the Caenorhabdites, they're highly derived, very evolved genus of nematodes. So the fact that they don't have eyes or opsins, that's a derived trait, not an ancestral trait. And the most common ancestor of C. elegans and other nematodes that have eyes and have opsins presumably also had eyes and opsins. So the Caenorhabdites clade lost their eyes and opsins. So maybe what happened with C. elegans and other nematodes potentially is that this ancient, highly conserved cellular stress response pathway was hijacked through selective pressures to enable the worm to more effectively discriminate good bacteria from bad so that it could survive better using spectral properties of pigments secreted by the bacteria, even though it had already lost in the lineage its eyes and its opsins. Because you're not going to evolve, you're not going to get opsins from the absence of opsins in any sort of short, evolutionary time scale. But you might be able to hijack an already light sensitive pathway that's still there to confer spectral sensitivity somehow upon it. So that's the evolutionary mystery that also, I think, is a fascinating question to address.
So if you're a worm, you don't necessarily need to see like an ant coming towards you because you're not going to go hunt that ant, but you do want to know what colored background you're on because you might be, in poison, so to speak, I guess.
Exactly.
Okay. Makes sense.
And the worms do have the ability to detect these toxins chemically. So, but as with all of sort of sensory biology, incremental increases in acuity, if you will, the ability to discriminate one thing from another can be powerfully selected for, even if they give subtle fitness advantages just based on the exponential mathematics, right, of natural selection.
And these seem to be somewhat innate responses. I mean, it'd be interesting. I don't know if you, TWiN, looked at whether you could train or associate one of a non-toxin with a variance of light, because that would be be really cool.
So yes, so the learning side of things is again a whole nother area to explore. Absolutely.
Right. Is there a significance to the blue amber ratios in nature? Does that make sense where the worms are actually living?
It makes a lot of sense, given that if you just look at the spectrum of sunlight that hits the Earth, basically there's sort of two major peaks of wavelength in the white light that hits the Earth. There's a big peak. I'm just talking visible, not UV. There's a big peak in the blue, and then there's kind of a trough, and then there's another big but much broader peak in the, sort of green, yellow, red region. And that's why if you look at the spectrum of the light sources that we use in the paper, the white light sources, what we call white light looks white to the human visual system. And the light bulbs that you can buy that are, quote, white light bulbs are that way because they look white to human beings. And it's because there's that kind of balance of intensity between the blue part and the long wavelength part of the spectrum. So given what light is available, it makes perfect sense. And this is what we do also as the human visual system, is distinguish blue, green, and red, is that you want to do your comparisons between the parts of the spectrum that there's plenty of illumination there to compare. So it makes perfect sense that the worms are comparing blue and amber because those are the relative peaks of the light that they're exposed to. Yeah. And then it makes sense. And another fascinating biological question is, why do so many microbes secrete such colorful pigments? these things are toxins and pigments, the same molecule? Is it just an accident that they're so colorful? Or is there some selective significance to the colors themselves vis-a-vis the function and physiology and ecology of the microbes? And then if you want to get into real fantasy land, you could say that perhaps what's going on is some sort of color-dependent arms race / manipulation of the behavior of animals by microbes by virtue of the colorful pigments that they secrete that alter the spectra of light that animals around them observe.
So I just want to throw out a red herring here. When I read this paper, I couldn't help but think of the dress. Are you guys familiar with the dress? Uh-huh. It made me think of that because it was a blue gold balance that, and some people's, I don't know, I can't remember if it was at what level it was occurring, but that some people tend to see blue more than the gold or the gold more than the blue and interpret it differently. I don't know that what is being described in this paper and what that phenomenon have anything to do with each other, but it definitely made me think of that.
It sounds like the final arbitrator for this is to put C. elegans on the dress.
See what they do.
Let C. elegans tell us what color it is.
I was also I know that we're not necessarily going like figure by figure, but so I'm going to just continue with that and ask, do we know any, like when in one of the later figures, so yeah, in figure 4, when he's doing all these genetic screens, what is the material that's used for that? Is that like whole C. elegans mashed up and everything's collected, or are there specific tissues that are collected? Because I'm just wondering, where are these, for example, the light, and I don't know if you guys say GUR3 or exactly. 3, are those differentially expressed in like, is there an access to C. elegans where you're going to have higher expression of those genes in one place versus another?
The genetic screen is done on intact behaving worms of different of the different wild strains that have been collected in various parts of the world. So it's a behavioral screen. That is,
right.
For each strain of worms, to what extent does it remain on versus leave the bacterial lawn in that particular spectral environment, either a blue-dominated environment or an amber-dominated environment. So it's a behavioral phenotype that is then associated with the genetic variation across these strains.
Right.
Is there any information? Do you guys have any information about where these proteins are expressed or anything like that?
So the light 1 and the GUR3, which have been explored previously for their roles in this kind of avoidance, escape responses to bright short wavelength light, there's some information about where they're expressed, and some of it makes some sense, and it's kind of confusing, and there's not a clear picture of the sort of cellular and molecular mechanisms by which those gene products operate to enable bright light driven escape responses. And there's even less known about the genes that Dupont identified, these other completely distinct genes that are in the cellular stress response pathways necessary for the spectral discrimination. No information as yet on that.
So we've kept you an hour now. Any other pressing questions from the group? I would like to know I assume you're going to continue some aspect of this in your lab. What are some of the interesting questions that you want to pursue?
So this is sort of an extra scientific point, but actually the goal is for Dupont to continue to work on this stuff when he starts his own lab. And so we won't compete with him on this. I'm not going to, and students and postdocs, the whole paradigm of like Deepan figured this out on his own. That's essentially how my lab operates. I don't tell people what to study. So students and postdocs who have come into the lab since Deepan, they got their own ideas. They're not interested in pursuing Deepan's ideas, and I'm not pursuing any ideas. I'm just creating an environment where people can pursue their own ideas. So it's he that will be doing this. But there are some obvious things to do, including such as was raised, like, well, how's any of this work? What are the pathways? And so the way that you do that, which C. elegans is so powerful for, is we have exquisite genetic control of cellular expression of trans genes and mutations. And so we can, very straightforwardly figure out in what cells and tissues is expression of these genes required to enable the color discrimination behavior. And then by figuring out what cells and tissues those genes need to be expressed in to serve this function, then that lets us get into a genetic hook of, okay, can we identify then suppressor or enhancer mutations that make the phenotypes either worse or better in those cells and tissues to start to work out the cascade, the pathway, the signaling, with the ultimate goal of identifying what specific molecules are absorbing these photons of different wavelengths, encoding the intensity of photon absorption at the two different wavelengths, comparing, right? There has to be a comparison somewhere in the nervous system of the activations by photons of those different wavelengths to then compute a behavioral output, which is increased or decreased propensity to leave the current situation of what bacterial lawn you're sitting in as a worm.
Yeah, this is a perfect scenario for starting your lab. I mean, this is what you would love to have is a completely new area to explore. There's no one else doing it, or not yet.
Not yet.
And
they will.
The grants basically write themselves because it's just obvious what the next questions should be, at least initially, and then you follow science. This is great.
Exactly. That's what you want.
So
that's what Deepan wants.
We had on another one of my podcasts, Daniel Cronauer from Rockefeller, who studies ants.
Yeah, I know quite well.
And they don't see, but they smell, right? That's what drives them.
Yeah.
So you don't have to, like you said before, you don't have to see if it's not necessary. If there are no paintings, you don't need to have eyes,
Yeah. I mean, this gets into the epistemology, if you will, of the organisms themselves. Ants and flies. These animals have a sophisticated enough nervous system that they can form a map of their spatial environment. And so flies clearly have a spatial map of their environment, and other insects for sure, bumblebees, etc., monarch butterflies. And they can orient themselves in space, and they have spatial working memory, and they can keep a map of what in their environment is in what places, and they can go to those places, or they can leave places that have things that they know need to be avoided. Best that we can tell as far as C. elegans goes, all they really can do, there doesn't seem to be any way that their nervous system could form a spatial map of their environment. And so what they basically, I think, do is on a just continuous basis, they compute a kind of cost benefit of, should I stay where I am and deal with what's right here, or should I go somewhere else and look for something better than what is here? And so they may have, certainly on very short time scales, they have to be able to compare one thing to another, or else they don't even know what it means to leave where they are. So they have to, at a short time scale, and maybe at a slightly longer time scale, they may be able, while not having necessarily a spatial map of where something good or bad was that they encountered slightly longer in the past, they may have the ability to at least remember that there was something better somewhere else. And that at least will tell them, like, I don't remember where, but I know it's not here, so let's move and go somewhere else. And so they're just continuously computing sort of the bias between staying where they are and consuming the food that's where they're at. If we're talking about trying to decide whether to just keep eating what's there versus going elsewhere, because it's not a binary decision, right? It's not that there's like bad food and good food. There's a spectrum of food from bad to good, and it can be very important fitness-wise for them to be able to discriminate more finely between better and worse sources of food.
I feel like humans need to learn from C. elegans. I mean, we have just as many problems determining, should I stay, should I go, what should I do with my life? Lessons in stoicism from C. elegans.
Yeah, unfortunately, not enough people know about them. It's true, though. All right, Michael Nitabach, Yale University. Thanks so much for joining us. Appreciate it.
My pleasure.
Thanks, Mike.
Thanks, Mike.
Thank you,
everyone.
Bye-bye.
Bye.
Bye-bye.
So I don't know if you guys know, but there's this great experiment in ants. So if you draw a magic marker circle on a piece of paper, the ants will not cross it because they smell the what's in the ink, right? If you knock out an odorant receptor in ants, then they go all over the place. They don't care because they can't smell anything, and that's what guides them. It's such a cool experiment. They made the first knockout of a gene in Daniel Cronauer's lab, and it was an odorant, which they identified by 7 transmembrane, and the magic marker experiment. So cool.
Yeah, this is making me think we need to get somebody who studies naked mole rats on here, because they're among those organisms, and you're like, how do they do stuff without all the same organs that we do?
Yeah,
or their close relatives. We
have a bias, right? Because we got what we have, and we can't imagine living without it. That's part of the problem, right? Mm-hmm. But I like this idea that they just die under the apple and a few are left. What a different way of living.
I mean, the thing that I find amazing is the social aspect of these insect colonies, where individual ants have behavior, but the emergent behavior from the whole colony is a different level. And I just can't get my head around how some of that can evolve, where it clearly needs communication between different organisms or different individuals. And at the individual level, the behavior doesn't matter as much, but it's the colony.
Yeah. Oh, well, they're highly social, right? And these army ants that Cronauer works on, the whole colony moves every night. The whole thing, these huge things, they just pick up and leave, and they go somewhere else. I mean, how do you get everyone to follow you, right? The other part is cool is that there are imposters in the colonies that somehow smell like the ants. They look totally different. Like you can see when the ants are moving there, if he has photographs of them, there are these big insects going with them, and they think it's an ant because it smells like an ant.
Yeah, so that evolution is absolutely mind-blowing. So Joe Parker at Caltech is working on this, and it's these beetles. So these beetles evolve both structural changes and chemical changes in their bodies to mimic the ants, and they're super selective for one kind of species. And he's been looking at how sort of the ancestral normal beetle, what's changed in terms of the genetics between these highly evolved specific parasites, essentially, in these ant colonies.
Very cool.
It's mind-blowing.
Do they eat the ants?
They don't eat the ants. They get the ants to feed them.
Yeah.
So they basically don't do anything. The ants think that these beetles are their colleagues and they're feeding them.
Vivianne, do you think humans should learn from them maybe?
Oh, absolutely. Yeah. I mean, I'm just seeing all of these opportunities for growth for humans from looking at these lower, quote unquote, organisms.
Anyway, that was really good. I enjoyed that very much. Really good stuff. Mm-hmm. Vivianne, before we leave, maybe since you are our newest member, maybe you could tell us a little bit about your history.
Sure. So I'll do as much as I can, the shortened version of my entire life story. So I was born in Northern California and left there when I was 16. Not totally by my own will. My parents moved, my family moved to Switzerland for my father's job. I managed to, and this is all like in when I'm like, I managed to just keep it together for 1 year before returning to the United States. That is said, now knowing that as an angsty teenager, I was just missing out on an amazing experience, being in a wonderful place like Switzerland. But anyway, I went back to the United States and I got my bachelor's in psychology at Bard College in upstate New York, which you wouldn't go to unless you wanted to be like an orchestra conductor or maybe an independent filmmaker. But they're trying to,
the president is a conductor, right?
Yes, Leon Botstein. Yeah.
And that's David Botstein's brother, guys. You know David Botstein, right? You've heard of him. Is he at MIT? No, he went to California. Amazing. Two brilliant people, right?
They are brilliant. And I mean, Bard was a great place they focus so much on like critical thinking and teaching you how to read in your first year that it really sets you up. So then I was doing some research at the end. I dabbled in some research at the end of my bachelor's, and I was it culminated in doing a literature review on the biological underpinnings of phantom limb syndrome, because I was very interested in neuropathic pain. And I did some experiments with zebrafish looking at kind of unchecked nerve growth factor signaling in the stump of an amputated fin, and how that could contribute to abnormal sprouting and things like that. So I was interested in research, but I was also maybe I should be a doctor, which several years later, when I was like, I don't want to be a doctor, my whole family was like, oh, thank God. We didn't know how to tell you this, but like, you don't even like people. Like, why would you want to be a doctor? And it's true that when I was in medical school, I went back after graduating from Bard, I went back to Switzerland where I had the opportunity to pursue medical school. And it is like what Jason experienced, where you have the same system in Switzerland where people who are interested in medical school from high school go directly into pre-med, and it's just like, 80% call rate. And the people who make it, they will go on to medical school. But so I had the opportunity to skip that first year. And it was really fascinating, but I was just getting more and more involved in, I was more interested in like the biochemistry courses and much less in the like how to take a good medical history. And I found that if I stayed in medicine, I was probably going to end up in a basement studying pathology. And I ended up in a basement studying other things as a scientist. But so when I decided, I spent a couple years in Switzerland, and then that's where I met my now husband. And he moved back to the United States, and I decided, I don't want to be a doctor, because I was also, alongside my studies, I was doing internships in labs, studying a variety of things, but largely focused on neuroscience. And then I decided, well, the person who I want to spend my life with is no longer here in Switzerland. I don't want to be a doctor anymore. I'd really rather pursue research. And so I moved back to the United States, to Washington, D.C., and got a job working as a research assistant in the zebrafish animal model shared resource at Georgetown University. And I did that not for very long before applying to graduate school. And I looked around at the different universities, which there are many really stellar institutions in Washington, D.C., and decided that the only place I wanted to go was Georgetown. So I applied to one school, and I said, admit me, or I'll just keep coming back. And they accepted me. And so even before I started, I think even before I was actually technically accepted, I was meeting with professors to see like what they, to express my interest in what they were doing and kind of decide where I'd want to go. And I met Jeff Wong, who did his postdoc with Robin Franklin at Cambridge in the UK. And he studies myelination. His PhD was in developmental myelin, just normal myelin. And then with Robin, he was studying remyelination following demyelinating injury. And so he had just started up his lab, like literally just started his lab at Georgetown. And so I said, I don't know what this whole myelin thing is, but, sounds cool. And I came to talk to him and he opened this book, like a textbook, and showed me an electron micrograph of a myelinated axon in cross section. And I was just blown away. I was like, I don't know if you guys have seen it. Probably, I imagine you have since you study neuroscience, but it's just an exquisitely organized structure. This nice,
it's like a tree with rings around it, like a tree.
Right. Yeah. So you see like a circle, which is the axon, and then there's concentric, really in the there are electron opaque, really dark circles around it, which is myelin, which is an extension of a glial membrane that has been lengthened and flattened and then compressed around the axon to just basically support the axon's function, which at some point we will talk about. And it was just beautiful. And I knew that this was the place for me. And that is where I ended up doing my doctoral work. And kind of what's come out of all of that is that I'm primarily interested in how cells decide what they're going to be. And in particular, cells in the central nervous system, glia. So I was studying oligodendrocyte development and how they arise from neural stem cells in the subventricular zone. And more specifically, I was studying how retinoic acid influences that. That I'm sure my research, I will expand on that at another episode, I'm sure. But so that's what I was studying there. And that kind of dovetailed nicely into, serendipitously into what I'm doing now. I'm at Vanderbilt University and I work with Dr. Bruce Carter in the Department of Biochemistry. And his lab actually studies studies in part P75 signaling and its role in peripheral nervous system development, because P75 is a neurotrophin that is a kind of switch between survival and death. And then additionally, he studies this novel receptor called JEDI, which is a mammalian homologue of the SED genes. Those are the apoptosis associated proteins that
Can you do that earlier without George Lucas suing you?
It's called JEDI-1. So it's JEDI-1. So I think we've been saved, but we're not it's not the only protein with that name, interestingly. So currently, I actually entered his lab because I was interested in, myelinating cells. And I thought, well, this might be a good opportunity to expand my knowledge to include Schwann cells, which are the myelinating cells of the peripheral nervous system. And at the time he mentioned that he actually had some very, very preliminary data. They'd been working on a JEDI-1 knockout mouse and the phenotype in the peripheral nervous system. And they had some very, very preliminary data suggesting that these knockout mice might have some phenotype in the central nervous system. And so I dove on that and am now looking at how this receptor, JEDI-1, which is an engulfment receptor, and it's involved in phagocytosing debris in the developing nervous system as those neurons die back, that its expression in microglia is necessary for postnatal neurogenesis from the subventricular zone. So it's a really fascinating project, which again gets back to what I'm interested in, which is how do cells become what they're going to become. So, if you're looking in the in the subventricular zone, the neurogenic niche, there are neurons, there are astrocytes, there are oligodendrocytes. They're all born from the same cells. How do those cells decide what to become and where to go? And then I think secondarily, a major interest of mine is how neural and non-neural cells interact. Because JEDI is also expressed by perivascular cells, so blood vessels. And in my work, when I was at Georgetown, the protein that we were studying was also expressed in perivascular cells. And so you have these expression of proteins in non-neural cell types, so vascular associated cells that are having big impacts on how the subventricular zone as a gliogenic and neurogenic niche functions. And so it's super fascinating. And I'm looking forward to, I just finished my first year. Half of that was spent at this desk at home because of COVID, but I'm really looking forward to the future. But so, yeah, that's me.
I know we haven't got time, but next episode we should also do this for Tim, because I don't even know where you grew up, Tim.
All right,
Hong Kong.
I'll do it next time. I thought we had, you guys, it was up to you guys last time because you had him on, so you didn't.
That was, yeah, yeah.
But we'll do it, we'll hit him next time because I have to go clip some mouse tails right now. So that is TWiN number 17. You can find the show notes at microbe.tv/TWiN. If we have lots of questions, we'll get to it at some point. But if you have a question or comment, TWiN at microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. Ori Lieberman, I don't know what to say. You've been at Columbia University, and you're going to change, but when you change, we'll tell people over on Twitter, Ori Lieberman. Thanks, Ori.
Thanks, guys. This was great.
Jason Shepard's over at the University of Utah. Jason Synaptic on Twitter. Thanks, Jason.
Yeah, thanks, everyone. That was fun.
Vivianne Morrison's at Vanderbilt University. Do you want to give out your Twitter handle, Vivianne?
No, not because I'm trying to be, secretive or anything, but I don't do the Twitter. I guess I'm more, I don't know. I'm just not good at it yet.
And Timothy Cheung is at New York University Medical Center here in the city. Thanks, Tim.
Thanks very much. Nice meeting all of you.
I'm Vincent Racaniello. You can find me at virology.blog. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll see you next time.